Welcome to our deep dive into statistical methods in bioinformatics. In this video, we'll explore how statistics powers discoveries in biology, from decoding genomes to understanding complex protein interactions. Bioinformatics sits at the intersection of biology, computer science, and statistics, making it a truly interdisciplinary field. As biological data grows exponentially, statistical methods become essential tools for extracting meaningful insight. Whether you're a student, researcher, or just curious about how data shapes modern biology, this journey will help you appreciate the crucial role of statistics in bioinformatics. Let's start by understanding what bioinformatics is and why statistics are so important in this domain. Bioinformatics is the science of collecting and analyzing complex biological data such as genetic codes, protein sequences, and cellular pathways. It combines biology, computer science, and statistics to interpret vast datasets generated by modern experiments. The field emerged as a response to the data explosion in genomics and molecular biology. Today, bioinformatics tools help researchers identify disease genes, predict protein structures, and understand evolutionary relationships. The integration of statistical methods ensures that the conclusions drawn from biological data are robust, reliable, and reproducible, which is vital for scientific progress. Statistics is the backbone of bioinformatics. With the sheer volume of biological data, traditional analysis methods fall short. Statistical techniques allow researchers to make sense of noisy, high-dimensional datasets. They help in hypothesis testing, pattern recognition, and drawing inferences from experimental results. For example, when comparing gene expression levels across different conditions, statistical tests determine which genes are significantly different. This ensures that findings are not due to random chance but reflect true biological phenomena. As we move forward, we'll see how specific statistical methods are applied in bioinformatics research. Bioinformatics deals with a variety of biological data types. These include DNA and RNA sequences, protein structures, gene expression profiles, and metabolic pathways. Each data type presents unique challenges for analysis. For instance, sequence data is linear and symbolic, while protein structures are three-dimensional. Gene expression data is often high-dimensional, with thousands of genes measured simultaneously. Understanding the nature of these data types is crucial for selecting appropriate statistical methods. In the next scenes, we'll explore how statistics adapts to these diverse datasets. Descriptive statistics are the first step in analyzing biological data. They summarize and organize data, providing insights into its central tendency, variability, and distribution. Common measures include mean, median, mode, variance, and standard deviation. For example, in gene expression studies, descriptive statistics help researchers understand the average expression level of a gene and how much it varies across samples. Visualizations like histograms and box plots are also used to reveal patterns and outliers. These summaries guide further statistical analysis and hypothesis generation. Differential statistics allow researchers to draw conclusions about populations based on sample data. In bioinformatics, this often involves testing hypotheses about gene expression, mutation rates, or evolutionary relationships. Techniques like t-tests, ANOVA, and chi-square tests are commonly used. These methods help determine whether observed differences are statistically significant or likely due to random variation. By applying inferential statistics, scientists can make informed decisions and predictions, even when working with limited or noisy data. Probability theory underpins many statistical methods in bioinformatics. It helps model the uncertainty inherent in biological systems. For example, probability distributions describe the likelihood of observing certain DNA sequences or mutations. Bayesian statistics, which rely heavily on probability, are used to update beliefs about biological processes as new data becomes available. Understanding probability is essential for interpreting results and making predictions in bioinformatics research. Hypothesis testing is a cornerstone of statistical analysis in bioinformatics. Researchers often start with a null hypothesis, such as there is no difference in gene expression between two conditions. Statistical tests are then used to assess whether the observed data provide enough evidence to reject the null hypothesis. P-values and confidence intervals help quantify the strength of the evidence. Proper hypothesis testing ensures that scientific conclusions are based on rigorous statistical reasoning rather than chance. Bioinformatics, researchers often perform thousands of statistical tests simultaneously, such as when analyzing gene expression across the entire genome. This leads to the multiple testing problem, where the chance of false positives increases with the number of tests. To address this, methods like the Bonferroni correction and false discovery rate (FDR) control are used. These techniques adjust significance thresholds to reduce the likelihood of spurious findings, ensuring that reported results are truly meaningful. Biological data often contain technical variations that can obscure true biological signals. Data normalization is a statistical process that adjusts for these variations, making datasets comparable across samples or experiments. For example, in RNA-seq studies, normalization accounts for differences in sequencing depth and gene length. Proper normalization is crucial for accurate downstream analysis, such as identifying differentially expressed genes or clustering samples based on expression profiles. Clustering is a statistical technique used to group similar data points together. In bioinformatics, clustering helps identify patterns in gene expression, classify proteins, or group samples with similar characteristics. Common methods include hierarchical clustering, k-means, and density-based clustering. These techniques reveal hidden structures in complex datasets, aiding in the discovery of new biological insights and the generation of testable hypotheses. Principal component analysis, PCA, is a dimensionality reduction technique widely used in bioinformatics. It transforms high-dimensional data into a smaller set of uncorrelated variables called principal components. PCA helps visualize complex datasets, identify major sources of variation, and reduce noise. For example, in gene expression studies, PCA can reveal clusters of samples with similar expression profiles, highlighting underlying biological differences. Regression analysis is used to model relationships between variables. In bioinformatics, linear and logistic regression help predict outcomes such as disease risk based on genetic markers. These models estimate the strength and direction of associations, allowing researchers to control for confounding factors. Regression techniques are essential for understanding how multiple factors jointly influence biological processes and for making predictions from complex datasets. Machine learning is increasingly important in bioinformatics. It involves algorithms that learn patterns from data and make predictions or classifications. Supervised learning uses labeled data to train models, while unsupervised learning finds hidden structures without labels. Applications include predicting protein functions, classifying diseases, and identifying biomarkers. Statistical foundations ensure that machine learning models are robust and generalizable to new data. Supervised learning is a machine learning approach where models are trained on labeled data. In bioinformatics, this might involve using known disease states to train a classifier that predicts disease risk from genetic data. Algorithms like support vector machines, decision trees, and neural networks are commonly used. Supervised learning enables accurate predictions and helps identify key features that distinguish different biological states. Supervised learning finds patterns in data without predefined labels. In bioinformatics, clustering and dimensionality reduction are common unsupervised methods. These techniques help discover new subtypes of diseases, group genes with similar expression patterns, or identify novel biological pathways. Unsupervised learning is powerful for exploring complex datasets and generating new hypotheses for further investigation. Sequence alignment is a fundamental task in bioinformatics, used to compare DNA, RNA, or protein sequences. Statistical methods assess the significance of alignments, distinguishing true biological similarities from random matches. Algorithms like BLAST and Smith-Waterman use scoring systems and probability models to identify conserved regions, infer evolutionary relationships, and predict functional elements within sequences. Markov models, HMMs, are statistical models used to analyze sequence data in bioinformatics. HMMs model biological sequences as a series of hidden states, each emitting observable symbols. They are widely used for gene prediction, protein family classification, and sequence alignment. HMMs capture the probabilistic nature of biological processes, allowing for robust analysis of complex sequence patterns. Phylogenetic analysis reconstructs evolutionary relationships among organisms or genes. Statistical methods estimate phylogenetic trees based on sequence similarities and differences. Techniques like maximum likelihood and Bayesian inference provide frameworks for tree construction and hypothesis testing. Phylogenetic analysis helps trace the origins of species, understand gene family evolution, and study the spread of infectious diseases. Gene expression analysis measures how genes are turned on or off in different conditions. Statistical methods identify differentially expressed genes, revealing how biological processes respond to changes like disease or treatment. Techniques include normalization, hypothesis testing, and multiple testing correction. Gene expression studies provide insights into cellular mechanisms and help identify potential drug targets. Microarrays allow simultaneous measurement of thousands of gene expression levels. Statistical analysis of microarray data involves normalization, background correction, and identification of differentially expressed genes. Techniques like hierarchical clustering and principal component analysis help interpret complex patterns. Microarray studies have advanced our understanding of gene regulation and disease mechanism. RNA-seq is a powerful technique for quantifying gene expression using next-generation sequencing. Statistical methods process raw sequence reads, align them to reference genomes, and count gene expression levels. Differential expression analysis identifies genes with significant changes across conditions. RNA-seq provides high-resolution insights into transcriptomes, enabling discoveries in development, disease, and evolution. Genome-wide association studies, GWAS, search for genetic variants associated with traits or diseases. Statistical methods scan the genome for single nucleotide polymorphisms, SNPs, that differ between cases and controls. GWAS relies on rigorous statistical testing and correction for multiple comparisons. These studies have identified thousands of genetic risk factors for complex diseases like diabetes and cancer. Predicting protein structure from sequence is a major challenge in bioinformatics. Statistical models analyze sequence features and evolutionary information to predict secondary and tertiary structures. Machine learning and deep learning approaches have recently achieved remarkable success, as seen in tools like AlphaFold. Accurate structure prediction aids in understanding protein function and designing new drugs. Biological systems are often represented as networks, such as gene regulatory networks or protein-protein interaction networks. Statistical methods analyze network properties, identify key nodes, and detect modules or communities. Network analysis reveals how genes and proteins interact to carry out complex biological functions, offering new perspectives on disease mechanisms and potential therapeutic targets. Metagenomics studies genetic material recovered directly from environmental samples. Statistical methods classify and quantify microbial communities, revealing the diversity and function of organisms in complex ecosystems. Techniques like diversity indices, ordination, and clustering help interpret metagenomic data. Metagenomics has transformed our understanding of microbiomes in health, agriculture, and the environment. Epigenomics explores chemical modifications to DNA and histones that regulate gene expression without changing the genetic code. Statistical analysis identifies patterns of DNA methylation, histone modification, and chromatin accessibility. These insights reveal how epigenetic changes influence development, disease, and inheritance, providing new avenues for diagnosis and therapy. Proteomics studies the entire set of proteins in a cell, tissue, or organism. Statistical methods process mass spectrometry data, identify proteins, and quantify their abundance. Techniques like clustering and network analysis help interpret complex proteomic datasets. Proteomics provides insights into cellular function, disease mechanisms, and potential biomarkers for diagnosis and therapy. Systems biology integrates data from genomics, proteomics, metabolomics, and other fields to model complex biological systems. Statistical and computational methods build predictive models of cellular processes, simulate interactions, and identify emergent properties. Systems biology provides a holistic view of life, enabling researchers to understand how genes, proteins, and metabolites work together in health and disease. Effective data visualization is essential in bioinformatics. Statistical graphics like heat maps, scatter plots, and network diagrams help researchers explore data, identify patterns, and communicate findings. Visualization tools make complex datasets accessible and facilitate hypothesis generation. Good visualizations are clear, accurate, and tailored to the audience, enhancing the impact of bioinformatics research. Reproducibility is a cornerstone of scientific research. In bioinformatics, statistical methods and computational workflows must be transparent and repeatable. Tools like version control, workflow management systems, and literate programming ensure that analyses can be reproduced by others. Reproducible research builds trust in scientific findings and accelerates progress in the field. Bioinformatics relies on specialized statistical software for data analysis. Popular tools include R, Python, Bioconductor, and specialized packages for genomics, proteomics, and systems biology. These tools offer a wide range of statistical methods, visualization options, and reproducibility features. Mastery of statistical software is essential for modern bioinformatics research. Integrating data from multiple sources is a major challenge in bioinformatics. Statistical methods combine genomic, transcriptomic, proteomic, and clinical data to provide a comprehensive view of biological systems. Integration enables the discovery of new relationships, improves predictive models, and supports personalized medicine. Effective data integration requires careful normalization, alignment, and statistical modeling. Scale of biological data presents unique statistical challenges. Big data in bioinformatics requires efficient algorithms, scalable statistical methods, and robust data management. Issues like missing data, batch effects, and computational limitations must be addressed. Advances in cloud computing and high-performance computing are helping researchers tackle these challenges and unlock new discoveries. Bioinformatics research often involves sensitive genetic and health data. Statistical methods must ensure data privacy, security, and ethical use. Researchers follow guidelines for informed consent, data anonymization, and responsible sharing. Ethical considerations are essential for protecting individuals and maintaining public trust in bioinformatics research. Statistical methods in bioinformatics are driving the shift toward personalized medicine. By analyzing genetic, molecular, and clinical data, researchers can tailor treatments to individual patients. Predictive models identify optimal therapies, minimize side effects, and improve outcomes. Personalized medicine represents a new era in healthcare, powered by advances in bioinformatics and statistics. Future of statistical methods in bioinformatics is bright. Emerging technologies like single-cell sequencing, spatial transcriptomics, and artificial intelligence are generating new types of data and analysis challenges. Continued innovation in statistical methods will be essential for interpreting these data and translating discoveries into real-world applications. The integration of statistics, biology, and technology will shape the next generation of bioinformatics research. Bioinformatics is inherently collaborative, bringing together experts in biology, statistics, computer science, and medicine. Statistical methods facilitate communication and integration across disciplines. Collaborative projects accelerate discovery, improve data interpretation, and foster innovation. The success of bioinformatics depends on teamwork and the sharing of knowledge and resources. Education in statistics and bioinformatics is vital for the next generation of scientists. Training programs teach students how to analyze biological data, apply statistical methods, and use computational tools. Lifelong learning is essential as new technologies and methods emerge. Investing in education ensures that researchers are equipped to tackle the challenges of modern biology. Open science movement promotes sharing data, methods, and results to accelerate discovery and improve reproducibility. Statistical methods play a key role in standardizing analyses and enabling data sharing. Open access to bioinformatics resources fosters collaboration, transparency, and innovation, benefiting the entire scientific community. Let's look at a real-world application: cancer genomics. Statistical methods analyze tumor genomes to identify mutations driving cancer. Techniques like differential expression analysis, clustering, and network modeling reveal the molecular basis of cancer and guide the development of targeted therapies. Cancer genomics exemplifies the power of statistics in transforming healthcare. Statistical methods are crucial in studying infectious diseases. Bioinformatics tools track pathogen evolution, identify transmission routes, and predict outbreaks. Phylogenetic analysis and genome-wide association studies help understand how pathogens adapt and spread. These insights inform public health strategies and the development of vaccines and treatments. Human microbiome is a complex ecosystem of microbes living in and on our bodies. Statistical methods analyze metagenomic data to characterize microbial diversity, identify associations with health and disease, and predict responses to interventions. Microbiome research is uncovering new links between microbes and conditions like obesity, diabetes, and mental health. Despite advances, statistical methods in bioinformatics face challenges. Issues include data quality, missing values, batch effects, and computational complexity. Interpreting results requires careful consideration of assumptions and limitations. Ongoing research aims to develop more robust, scalable, and interpretable methods to address these challenges and improve the reliability of bioinformatics analyses. In summary, statistical methods are at the heart of bioinformatics, enabling researchers to extract meaningful insights from complex biological data. From gene expression analysis to personalized medicine, statistics drives innovation and discovery. As technology evolves, new statistical challenges and opportunities will arise. By embracing interdisciplinary collaboration and continuous learning, the field of bioinformatics will continue to transform our understanding of life and improve human health.